It's my pleasure to introduce our last speaker for today, Dr. Jessica Whitehead from Harvard Medical School and Brigham and Women's Hospital. Thank you.
Actually, I really want to thank Mike for organizing this and the Allen Center for having me and for supporting some of the work I'm going to tell you about today. And I've since moved the lab to Harvard University, so I also have to thank them for their support. It's really fun to be the last talk at the symposium, and I really want to give some perspective on this field about what are some of the remaining challenges, but also the opportunities in really understanding how complex tissues can be functionally regenerated with perfection. So the first half of the talk will be some phenomenology, which has been long-standing, and then we're going to transition to, with the advent of new tools in this system, we'll just transition to some of the ways that we can get at mechanisms now as a field. And so first I want to just get you thinking about this question of regeneration. When an animal first develops a structure, such as a limb, this ordinarily happens at a prescribed time during development. And for a given species, it will be a fairly given size or a prescribed size. And usually, the structure is starting from undifferentiated cells. So these are sort of cells that are allocated during the normal developmental life cycle, and they're meant to go and do this particular job at that particular time. However, when we contrast this to regeneration, we have to really think about these other variables that are on the scene. The animal could experience limb loss at any given time in its life. So axolotls live about 10 years in the wild, and this could happen when the animal's 9 years old. Of course, the animal is going to be of varied size. So when the limb initially develops, it's much thinner even than a toothpick. When an adult axolotl has to regenerate the limb, it's about as thick as my pinky. So these are thousands of times more massive than the original limb. And then, of course, you're also starting from a different pool of cells than what existed during normal limb development. And so these are all challenges that the animal has actually overcome. And they're also things we need to think about when we're thinking about someday applying this information therapeutically. This is the model system that we're studying. Many salamanders can regenerate limbs. We choose to study the axolotl because of many genetic tools that are now available in this organism, but also they have a relatively short generation time, which is still 1 year in the lab. And so what I would like to argue is that much like an IKEA manual for putting together a complex piece of furniture, these animals have at their disposal all of the tools, all of the parts, which we'll talk about later. They have to execute this in the right environment, so the cells have to be in the right environment to make this happen. And then finally, most importantly, they have to have the instructions. So without the instruction manual, you won't be able to construct this limb. And the animals have all of this information innate to their body. And so together, these things can achieve the regeneration of a really, truly complex system. And if we think about the axolotl limb, as shown here, inside this axolotl limb are all the same tissues and cells that human limbs have. So muscles, cartilage, bone, nerves, blood vessels, skin. We could go on. And if you just look at the pattern of these limbs, in this case, I've stained this limb with 2 dyes. The blue recognizes cartilage cells, and the purple recognizes bone. You can appreciate that they're morphologically quite similar in their pattern to human limbs. And they also undergo this kind of skeletal development or skeletal regeneration, much like human long bones develop in the first place. So endochondral ossification is illustrated by the purple emanating from the middle of the long bones. And we can compare this to the human arm shown here. So we think this is a really nice model to aim for to understand sort of what are the instructions for regenerating this structure. And shown here is an axolotl from our colony that went to live in a videography studio. And I just wanted you to see this video so that you can appreciate a couple things. I'm only going to point out 2 things here. So the first thing is that it's going to get an amputation fairly close to the armpit up here, and over the course of 8 weeks, a little bump forms. And this is sort of where all the magic happens. This little bump has a name. It's called the blastema. The blastema is a structure where the progenitor cells for the new limb go to proliferate and then get repatterned and form the limb. And the other thing I want to point out is that at the end of this entire process, you can't tell which limb was amputated and which one was never even injured. It's that perfect. And in fact, at the very end, another thing that we really have to recognize is that there is 0 scar. So you can't even see a scar in these animals. In fact, they generally heal all wounds without scarring, which is a totally fascinating separate topic that we can talk about later, but also connected to limb regeneration. And so how does this happen? People have known about this happening in salamanders for more than about 250 years. But it's been a really tough problem to crack because the appropriate molecular tools just weren't there yet. But what I want to show you now is basically the cartoon schematic of how this happens on a more gross anatomical level. First thing to appreciate is that no matter how much of the limb is lost, just only that part grows back, right? So in this case, we have two newts. And the one on the left was amputated more distally. So this is the distal to proximal axis on a limb. And you can see here that this one basically lost just its hand. Meanwhile, this animal lost its entire arm. And in about the same amount of time, this animal grows back just the hand, and this one grows back an entire arm. So we have to know how that happens if we want to harness these kinds of clues for regenerative medicine in the future. And if we think about the main morphological steps, then these are shown here. So you can see that following amputation, very soon thereafter, wound epidermis forms. So this forms via the migration of epidermal cells that remain on the stump to cover the wound. And it starts very quickly, within minutes or hours following the amputation. And we know this wound epidermis is especially important for limb regeneration because you can block its formation, and then the limbs fail to regenerate. Humans can regenerate digit tips, and the same is true in the human condition with children whose digit tips have been lost. If they're suture closed in the ER, they also fail to regenerate. But if you leave them open, then they do regenerate digit tips. The next thing that happens is that the visible blastema forms, and that's just directly underneath this wound epidermis. So at the tip of the stump underneath the wound epidermis. And in some ways, this is sort of akin to a niche in the stem cell world, or a transient amplifying population even, except for it can be executed on command. So the animal grows this when it needs it, and then it resolves it, goes away, and then it can grow another one potentially in the future. Shown in gray are all the new parts of the regenerated limb. And while these questions out here are super fascinating, so reconnection, for example, or scaling the limb or when to stop, largely my lab is focused on how do you make a blastema, because we know the mammalian situation goes awry at these early stages. So some of the questions we have tackled are, what happens to make a blastema? What's the interaction between the wound epidermis and these activated progenitor cells? What are the genes that are driving this process? Where the cells come from? I don't have time to get into all of the logistics about the different cell types that are present in the stump and who gets activated and where they go. But we have this sort of gross schematic here showing that some cells, probably for the most part, tissue-specific progenitor cells, but there is documented some small amount of transdifferentiation that occurs, and how much can occur is actually an open question if you push the system. But some cells get activated post-amputation, and then they're required to migrate to the wound site where they proliferate like crazy under the influence of potentially mitogenic factors from the wound epidermis, so they grow the blastema bigger, and then it goes on to repattern and form the new limb. It's possible that once you get a blastema, this could be akin to a giant limb bud, which is the structure that gives rise to the original limb in the first place, as far as like setting up the axes and these kinds of things. This is possible. If in that case, I'd like to argue that these early steps are really critical because, as we discussed earlier, we're starting from different cellular material than we would be in an embryo, or in this case, a hatchling, which is when they grow their limbs. Another important thing to recognize is that there are blood cells in this equation. I've shown red blood cells, but actually there are a lot of white blood cells also. And the immune system is documented to be very important for the process of regeneration. However, it's very poorly understood what its role is in this process. Okay, so how can we characterize the autonomy of the blastema? This has been an old and important question, and there's been some really nice, elegant experiments, transplantation experiments done several decades ago that address this question. And the sort of conclusion from these experiments is that once the blastema has grown to be a certain size, or a certain age, a certain level of maturity, that it can be transplanted and has all of the rules and basically all of the materials and the instructions to make what it was supposed to make. And so how do we know this? If you take a mature blastema from an animal that has undergone a proximal amputation, as shown here, and you transplant it to another area of the body that's receptive to regeneration, so in this case, the flank of the body, what you can get is a totally perfectly formed limb coming out. And note that since this was a proximal amputation, an entire limb grows, shown here. However, if you transplant this blastema to another area, so you might say, well, we would expect that limbs would grow out of this area because it's in between two limbs, and this is the flank area. It's been shown in development that if you give certain FGFs there, that you can induce the formation of an ectopic limb. What if you put it someplace else? If you put it in the eye socket, for example, what can happen is that you can grow a limb out of the eye. And so this is pretty cool, and it really sort of underscores that once the blastema is mature, it is quite autonomous. We don't really know all the rules going on inside there, but we'd like to know them. Another example is if we consider how much of the limb will grow back. So I just showed you that if you take a limb from a proximal amputation, it'll grow the whole thing back. And this really nails that question even further. So if you take a donor blastema from an animal that underwent a proximal amputation and you graft it to another animal's proximal amputation, so we have two green blastemas here, they're both proximal, then the animal grows an entire limb from the donor blastema, as shown here. And note that it actually emanates from this proximal location also, so that's pretty cool. It's even cooler when you think about this, and that's that if you take a distal blastema and you graft it, again, onto one of these proximal amputations, that distal blastema will only grow the hand, because it was programmed to grow the hand, and interestingly, it grows it where the hand goes, not up here by the armpit. So this is super cool, and we're still trying to figure out as a field what are the rules that govern these kinds of behaviors. We do know, though, that there is some way to overwrite this information encoding, and there's been a chemical that's been demonstrated to do this. So this is a really beautiful experiment. This was published by Malcolm Maiden several decades ago, and you can see that ordinarily a distal amputation gives rise to just a hand, but you can reprogram that tissue and force it to grow an entire arm, including the elbow joint, for example, if you treat this animal with retinoic acid during the process of regeneration. So we know it's possible to overwrite the instruction manual, but we also want to know what's the endogenous instruction manual and how to harness the information inside it. This is an example of a really sort of novel way to do this, to ask the animal to overwrite the instruction manual, but for which we are still trying to figure out how this works. In this case, if you amputate the hand of a newt and you implant it into the coelomic cavity, what will happen is that essentially no regeneration will occur. So they don't grow a hand into the body cavity. And if you let this heal and give the animal some time and then you amputate the limb, then you might ask yourself, well, what's going to happen to this structure? And what does that tell us about the biology of limb regeneration? And you can think that there's two possible outcomes. And the first outcome, we'd probably all agree that this top arm is going to regenerate. But the bottom one is what is questionable. And you can see in this first outcome, perhaps it would just heal over and remain a stump. And in the second outcome, it would grow another arm. And what has been observed is that the second outcome happens. And so this is super cool when you think about the fact that what the animal has just done is over the course of this amount of time that it's been connected up in this sort of artificial manner, it's reprogrammed the proximal-distal axis for that second arm there. And we'd really like to understand sort of the code for how this happens. And when we think about the autonomy of the blastema, another thing that we might want to consider is what we talked about earlier about how when the animal is first developed a limb, it's very tiny. And those tiny limbs also regenerate. But if you cut a tiny limb off, it grows back a tiny limb. And if you cut off a really big limb in a mature animal, it grows back a really big limb. How the heck does that happen, right? Some clues have been gleaned from this really beautiful old experiment in which there are two different size species of salamander. So these are two different species, and some modern pictures are shown here. This one is about twice as large as the top one. And just like in many embryological experiments like quail, chicken, chimeras, et cetera, you can do these sort of cut and paste experiments with salamanders, and they take grafts really well. And so what these people did was that they swapped the limb field during embryogenesis. So this would be sort of like the precursor cells for what's going to form the limb, and between the two species. And then they allowed those salamander embryos to grow up, and then they asked, OK, now that I put the limb field from a big salamander onto a tiny salamander, is that limb going to resize and become tiny because it's on the tiny salamander body, or is it going to grow big, and vice versa? And so this is illustrated here, and this is about the age and axolotl world that the experiment was performed. Again, this is limb development question, not regeneration. And what they found was that in the tiny species of salamander, the grafted limb, in fact, grew very large. And so this illustrates that this information about to grow a limb was taken with it, but also the size of the limb. So the size of the limb matches the donor size, and they observed the same thing in the large animal. The large animal grew a tiny limb from the donor. And so what this really shows is that it's definitely the case that during normal development, this information about size is encoded in these precursor cells. But what we wanted to also interrogate sort of is this idea about how this is captured during regeneration, or how this operates during regeneration. And one of the experiments that we've done in my lab has touched on this, and this experiment was originally designed to ask this question is, does the limb need a reference, like a reference model to regenerate? Or another way of looking at this is, can you challenge the animal in the adult state to grow the limb for the very first time? So this is a question that's sort of uniquely accessible in this kind of an organism, because they do regenerate limbs even as 9-month-old adults. And so what we did was we took these are hatchlings, very tiny axolotls, when they had their first limb buds, and we just snipped them off, and we kept snipping them off once every 10 days until the animals were 9 months old. And then we said, okay, now can you grow a limb? And we were curious to see what would happen. And so we did this many times, and we waited for the animals to grow big, and then we asked what happened. And what we found was that in the control animals, so these are the siblings from the same clutch, these animals lay hundreds of eggs and one mating, and so we would always reserve some controls, and you can see this one here. This would be like the size that it should be at that age and the size of the body. And we saw in about half of the outcomes there were no limbs grown at all. So these animals couldn't grow limbs, and you could argue that they needed some kind of reference. What that looks like mechanistically, we could talk about that later, ways to address that. So do they no longer have progenitor cells, for example? But in the other half of them, what we got were miniaturized limbs. And this is super surprising to us because it's an example of having decoupled the appendage size from the body size. And this is actually a fundamental question in biology for which there's really not a lot of information known at this time, which is how do you take an animal and it's supposed to grow up to be a given size, and how do you actually scale those appendages to match the size of the animal? What was super cool about this is that these miniature limbs, their growth plateaued. So this isn't just that they're in this longer growth phase, and we could document that in the laboratory. But we also challenged them to regenerate their limbs and ask, would they now resize according to their body and their age, or would they grow back a miniature limb? And what would that tell us about the programming of these blastemas? And what we observed was that in every instance of a miniature limb, a miniature regenerate grew back. And so this condition is permanent, and it's basically like permanently resetting the size of the appendage to the size of the body. And we're still working on the mechanisms controlling this phenomenon, but we think they'll be really exciting to explore when we're thinking about body plan and then also about information transfer between extremities and the rest of the body. Another question that's super salient in this field is, why doesn't the blastema act like a tumor? We don't really have time to get into tons of data about this, but in some ways, as I told you earlier, the blastema arises from the activation of progenitor cells, and these can be either stem cells or possibly, in some species, dedifferentiated cells, and they accumulate and they proliferate like crazy. So this is very similar to what can happen in a tumor, but why does the animal not grow a tumor and instead it grows a proper limb? One of my favorite experiments that addresses this question was done by Charles Breedis, and he gave newts chemicals that cause cancer in mammals. And he asked the question, do you get a tumor or do you not? And what he observed was that you very rarely, almost never, do you get a tumor. Instead of getting a tumor, often what you get is an ectopic limb. So these animals take these kinds of cues that would ordinarily cause tumors in mammals, and they direct them towards a patterned outgrowth. And some of the work that we've done with the Allen Center, which I'm not going to get into today, addresses some of the molecular mechanisms that are at play that allow blastema cells to proliferate while maintaining genomic integrity. So this is a challenge in vitro for regenerative medicine applications of cultured human cells. Is like when you have cells that are very highly proliferative, you have to monitor whether or not the genome integrity is still intact before you would, for instance, put them back in patients. And in the axolotl, the same thing is operational, but during normal limb regeneration. So we've detected an upregulation in the DNA damage response in these blastema cells during normal limb regeneration, and we've also identified factors that are required for preventing DNA damage accumulation during the process of regeneration. And now you can make a knockout in an axolotl. We've knocked some of these genes out and shown that they're required for normal limb regeneration. So as I just said, highly proliferative cells are prone to DNA damage and errors, and that if we have broken DNA, that can lead to tumors. However, salamanders turn on the DNA repair pathways to sort of quench this damage. And they anticipate that this is going to happen, or their cells anticipate this will happen. And so these things are turned on during the normal course of regeneration, but you can break it. OK, so part of our work in the Allen Center is really investigating this broader question. And that's how it's the broader body plan information encoded in individual cells. So this is a big question, and many people here touched on this in the presentations that they gave. And this is something that we're currently addressing with axolotls. And in the left is a GFP axolotl, so all the cells express GFP constitutively. And that can be used as a source of donor tissue, and then therefore you can track the cells around, right? And if you take a tail from one of these animals and you amputate it, this leads to a tail blastema, shown down here. They can also regenerate tails in many other body parts. If you transplant this onto a freshly amputated limb, then you can track these cells and ask questions such as, does this blastema give rise to a tail or a limb? From the autonomy work that I described to you earlier, you would predict if you did this perfectly with a really old blastema, you would get a tail. If you did it with a young blastema, you might get a limb. In between, you might get something in between. You can ask the question, what do you get? Indeed, we have seen a spectrum of outcomes. In some cases, the morphology is decidedly tail-like. Sometimes it's in between, and sometimes it's almost like a perfect limb. And so this one actually did go on to make a relatively perfect limb. This is just sort of the in-between state that's photographed here, 1 month post-transplant. So you can see the digits are starting to form there. And you can track the cells and see and ask questions about what structures have they contributed to. You can sort them out later, and you can go and compare, for instance, their transcriptomes to adjacent cells, et cetera. You can do this with a red animal and a green animal so that you can track both cells with a positive marker. So in this example, this is another example of that exact same experiment, except for we have a green host and a red tail blastema. And you can see here that this thing is in the process of making something limb-like. However, if you look into the patterning of those cells within that limb, you can observe that the patterning of those cells is decidedly tail-like. So these are definitely muscle fibers, but they're exhibiting a tail-like morphological pattern or tissue pattern. It doesn't look anything like the muscle fibers that would be observed in a limb. So we're really curious about using this platform further and to ask questions like, do these red cells still identify as tail or not? Do they instruct the limb-derived cells to behave differently, et cetera? OK, so these are some of the phenomenology questions that are still out there that we're starting to tackle. But what I really want to also leave you with is that these are very tangible questions that can now be answered. And the way that I want to transition to this is just to give you the flavor of the work that my lab has done to identify specific genes that are required for normal regeneration. Because once we've shown that you can do that, then you can apply the same techniques to some of those more nuanced and sophisticated questions that we were just looking at. And so something that we did very early was to say, we really don't know how blastemas are built or what drives their function, so let's just take a more agnostic approach to figuring that out, and let's just do a transcriptome study. And I'm not going to get into all the details, but we sort of made an atlas, a tissue-mapped reference transcriptome from all these different tissue types, and then we compared them to the gene expression in blastema cells. And of course, we generated a lot of heat maps. This was done in collaboration with the Broad Institute. And what you can see here is I'm just plotting in red is high gene expression. So these are the putative blastema markers. There's about 150 genes that were very highly expressed by blastemas versus all the other tissues we sampled, including unamputated limbs and the presumed parental tissues that give rise to blastema cells. So we took the very simple hypothesis that some of these genes shown over here, since they're very highly induced and some of them are quite specific markers like this KZALD1 gene, would be important for blastema function. And so what we've been doing since then is validating these genes and then going on to knock them down and then knock them out. So for knockdown experiments, we usually start with a morpholino. If there's a chemical inhibitor, we can also use that of the protein activity, for example. But in the meantime, we have to cook the sort of longer-term CRISPR experiments because, as I told you, the generation time is quite long. And so it takes a couple of years, for instance, for a student to get a true homozygous null animal. And so for this gene, we actually now do have true homozygous null animals. And we're waiting for them to get old enough to address the question of limb regeneration in adults. So all this work was done in adults. And this gene is a really interesting gene because it encodes a secreted protein. And this secreted protein has an insulin binding domain, and you can see that it's very highly induced. This is an RNA in situ hybridization, specifically in the blastema, not in the wound epidermis, and not in the stump. And we were able to show by knocking its expression down that it's required for blastema growth. And so you can see here at this intermediate time point, when the control limbs have attained this much larger regeneration, and they have 4 digits shown here. And the knockdown ones, they have just a blastema at that point. And if you were to fix and stain these limbs, you can see that the cartilage pattern is largely already laid down in the control, whereas it's barely even started here. And so we think this is one factor that we'd like to know more about. Its activity during normal limb regeneration. Another one we identified in the same study was CIRBP. So this is the cold-inducible RNA binding protein. You might have noticed on that heat map I showed you that everything that was highlighted in yellow is a predicted RNA binding protein or a protein involved in RNA life cycle. So we predict that probably there's going to be a lot of RNA life cycle regulation going on during the normal process of limb regeneration. So after validating this, we knocked it down, and we saw a different phenotype than what we saw with the knockdown of KZD1. In this case, what we saw, we also saw a slowing in blastema growth, but it wasn't due to cellular proliferation. Instead, it was due to excessive cell death in the CIRBP knockdown condition. And this really tells us that this molecule is required to protect blastema cells from dying, which is also something that you would postulate would be required from a therapeutic standpoint, or even just for normal animal regeneration standpoint, is that following amputation, it's a very hostile environment. Cells are sometimes disconnected from their supply of nutrition, for example, and they may need specialized factors to keep them from dying. Okay, so I'm not going to get into this, but of course we have drilled down to the single cell level, and this work is now published, so you can take a look at it if you're interested. We've characterized gene expression in individual cells from blastemas, but also from reference tissues. So these guys we did at several different time points: wound healing, mid-bud blastema, early blastema. And here we did the unamputated limbs. So you can imagine that if there are resident progenitor cells here that share some gene expression profiles with blastema cells, that we'd be able to capture that, and those are topics that we're looking at now. We also can compare these individual single cells derived from blastemas, for example, to the bulk tissue sequencing that we've done in the limb. This is just one example of a gene that we pulled out. So when we devised this study, we decided to take not just blastema cells, but also the wound epidermis, because it's very poorly understood what the role of the wound epidermis is, except it's known that it's absolutely required. And so we wanted to find not just more markers, but also more candidate genes for controlling wound epidermis function. And this is one example of a gene that is very specific to wound epidermis, and it comes on very early. It's a specific kind of keratin that we're interested in its role, not just extracellularly, but possibly also intracellularly. And of course, I could show you all of those kinds of t-SNE maps and those kinds of things, but you can look at the paper instead. But what I wanted to leave you with from this study is that there are tons of different cell types that we've identified during normal limb regeneration for which we basically have no idea what they're doing. And one thing that we can highlight are, for example, many of the immune cells. So it's been shown in a beautiful paper by James Godwin that macrophages are required for limb regeneration. So if you poison the macrophages, limbs can't regenerate. If that lineage replenishes, then that same limb can regenerate. But what are some of the other immune cells doing? We don't know the answer to that question yet, but I have a great postdoc who's working on that question right now, and he's trained in immunology. We can also address some of these older questions with these kinds of studies. So how is this information encoded that this proximal blastema grows a whole limb and this one just grows a hand? In the transcriptome paper, what we did was to compare proximal and distal blastemas to one another, and you can see that we identified candidate genes that would be great candidates for imparting this kind of information. We're currently testing those. And highlighted here is one of the lead candidates, which is a Hox gene called SHAX. And we predict that we may possibly be able to reprogram distal blastemas to proximal by overexpressing this. OK, so the last thing I want to talk about really quickly is some of the stuff that's like the most pertinent to the collaboration with the Allen Center, and that's to look at this question of bioelectricity. So how can bioelectricity control patterning outcomes during limb regeneration? We know that it's required for limb regeneration, that there's some influence of it during normal limb regeneration in, for example, this newt. So this is a beautiful paper that showed that there's a current efflux that comes out of the stump following amputation in newts. And what Richard Borgens also did was to reverse that current efflux using electrodes and show that if you do that, so basically you block it, then what happens is that these limbs basically scar over and they don't regenerate. And so this was a really, sort of seminal paper that showed that you can manipulate bioelectricity and grossly affect limb regeneration. But we wanted to look at this a little bit further. And so what we've been doing is using both drugs, but also overexpressing particular ion channels and gap junctions. And you can do this by making transgenic animals, but as you might imagine, overexpressing things like that can sometimes perturb development. So for many of them, we've turned to this retrovirus that I developed its application to axolotls while I was a postdoc. And so what you can see is that if you infect blastemas with this integrative retrovirus, you can get a high amount of expression. And that limb I showed you at the beginning of the talk that was super green, that was not a transgenic. It was a virus-infected blastema that was allowed to grow out. And you can also infect limb buds. And this work is done by a super talented postdoc, Kostas Sissounis. And what he's been able to show is that if you overexpress particular ion channels, you can grossly disfigure the regenerate, as shown here. So here we have a couple of potassium channels and a sodium channel, and you can see that in each case, particularly what's called the autopod, so the distal element of the limb, has not properly regenerated, and this is highly significant. So we want to know, on the flip side, this is overexpression. We also want to know if these things are required for regeneration, so we're doing studies now to look at that. And he had an even more specific phenotype when he overexpressed this particular gap junction. And the phenotype was very discrete, and it's always syndactyly, so that's the fusion of the digits. And you can see here is that in both the forelimb and the hind limb, when this connexin 26 gap junction is overexpressed, there's a high amount of syndactyly shown here. But the limbs largely regenerated just fine. But they had a patterning defect, which we think is super interesting. And the data is shown here was quite significant. And so what we want to do is ask this question, does blocking these activities also impair regeneration, and how does bioelectricity influence cells to drive shape? So I would argue that now is the time to completely decipher this instruction manual for limb regeneration. The animals have the tools, and now we have the tools to interrogate them. We want to know more about the parts, so the cells and the genes, the environment. So we didn't get into this, but we've touched in the lab on some of this environment issue and about anti-scarring and how that contributes to a pro-regenerative environment. And then the instructions are the blueprint to make a perfect limb. And if we knew how this happened, then we might be able to, as a field, scientifically approach this question better in a human being. So if you were going to make a human arm or leg regenerate, you would need some activated cells, whether they come from the patient, which would be, sort of like a holy grail, or they come ex vivo. Those cells are going to need some cues for promoting growth and survival in a hostile environment, and preventing tumors, because you wouldn't want to trade regrowing your hand for cancer. And you want to make sure that they're in an environment that promotes regeneration instead of scarring. And then finally, the subject of this entire meeting, which is that these cells need patterning information to create an exact replica if it's going to be useful. So finally, I want to thank Kostas, Nick, Donald, Kimberly. Some of these people were the ones that did the most of the work that I showed you earlier, and then our funding sources, and of course the Allen Center, and also Mike Levin and Josh. And I can take any questions.
Questions down here in the middle?
What's the current understanding about innovation of regenerates? So particularly where it's in the wrong location or where you've disfigured the limb in some way.
Do you mean, so are you thinking about some of these old experiments where you add an ectopic bone and then amputate and you get a third?
And muscles as well. So
right,
muscles innovated, how are they
right.
innovated?
So my current hypothesis about that is that those are prime examples of sort of resetting what Mike would, or we could postulate as a field, the target morphology. So once you've reset that, then if given enough time, then the animal's tissues have established new coordinates for who's going to go where. And that sort of Napoleon experiment, I think, would also support that idea. Do we know how that happens? No. But I guess I would also postulate that it would be time dependent, so like how long you left.
But I guess the
material
question
in.
is, do you know that it's functional? So I guess,
You mean like it's integrated and functional?
right. So if you've got a third limb growing out of your ribs, is it innervated? Can you walk on it?
Okay, right, so that's a great question. So we haven't really done any, for instance, like behavioral assays or, stainings to check how many neurons are in there or where the cell bodies are, et cetera. But, if you wanted it to be functional, it would have to hook into the nervous system. It definitely hooks into the circulatory system. So you can transplant an entire tail to the flank. And we did this, and we did many of them. And that will revascularize, and then you'll just have a tail there. If you cut it off, it actually still remains a tail. So it definitely integrates circulation-wise, but as far as integrating nervous system, I would be hesitant to conclude anything at this point. Yeah. But apparently that's sort of where the line is as far as like what's considered extra for the human being. So like, for instance, I've heard that the DoD is not going to support the idea that people in general would be pursuing extra limbs for battle or something like this, right? That's like beyond the scope of what, is that what you were asking? Yeah, that's like unethical, yeah.
Oprah's program, there was a man who had, the tip of the finger grew back.
Yeah.
Do you have any updates on that or what that's all about?
Right, so I remember that man because in the medical literature, it's only documented up to the age of like 13 or 14, but anecdotally, many times people who are much older say that they've regrown their fingertip, like in their 20s or maybe 30, right? But this guy was special because he, I think, was 60-ish or so, and he had this pig's bladder extracellular matrix, which they were calling pixie dust at the time. I think this came out of Pittsburgh. I don't have an update on that. I wouldn't rule it out. I'm not like so. I think the jury's out on that. Do I think that sprinkling extracellular matrix on, that was chock full of like BMPs and FGFs, for example, could stimulate a pro-regenerative response more than what you would get without it? Of course, that's possible. I think that's possible. But I haven't seen anything where they've like taken that particular one much further and like, gotten it much more specific about what caused that outcome. Yeah.
Right there in the middle. Thanks.
In the experiment where you were growing up the axolotls to try to get them to have the small limbs, so you kept cutting them.
Yeah.
So for the ones that didn't grow back limbs, did they just like give up on their limbs growing, or did you just?
Right. So we kept them for a very long time, and they never just spontaneously, grew a limb. But I'd like to repeat this experiment and then do this swapping style experiment, but within an axolotl, and then to address the question of regeneration and also within a species. But no, I don't know. They didn't do anything after that. Yeah.
In the back middle.
Right, so picking up on that same experiment, it seems in order to ask the question of why they stop at a particular size or reach a growth plateau at a particular size, you seem to have to have a model for why they stop at all. Mm-hmm. If they're reaching a growth plateau and they're doing it extremely reliably in normal development on the two sides, there must be some information that the tissue has about size.
Right, I don't disagree with you. I don't know in the axolotl like how they're calculating it, but we could even take this question one step further and ask how come your left arm and your right arm are the same size. And for people who are interested in that, I would point you to this recent paper by Alex Joyner's lab, and I think it was in PLOS Biology ultimately. It's beautiful, and because there are promoter constructs that can be used transgenically in mice just on one side versus the other, they could ablate like cartilage cells, for example, in the left limb of the mouse, and then look and see that the right limb would actually compensatorily regulate and change its size based on that. And they tracked it down to some circulating factors. I think it was insulin in this process. And so I didn't really get into this. We also have some experiments that have been looking at systemic factors during regeneration, because when we amputate a limb, there's this proliferative response, right? on the limb that is amputated, but it's also throughout the body, and the other limb, but also the heart, the spinal cord, the liver, for example. And so I'm very interested in how that connects to, for instance, the work that we just talked about. And I think that it's an open question, but she did a nice job in their paper.
Okay. Thank you so much.